Okay, what I wanted to say, so we have learned basics of biodiversity, and in biodiversity we have learned species diversities, number of species and abundance come under the species richness, and we have used diversity index, uh, that's Simpson index or diversity index to calculate it. And also we learn unit 2 a little bit about the quadrant method. Here, this is separate practical now, and we are going to learn these practicals uh, to count number of species in an ecosystem. Okay, so it's like this. If there is a area, and if you want to measure the biodiversity of a particular species, we have to do a ecological survey. So we are going to count the number of species in this area. Now think this complete area, the area of this ecosystem is something like from this direction, let's say like 5 kilometers, and maybe this direction again 5 kilometers, and this direction maybe 3 kilometers. So we can understand it is easily around something close to 20 square kilometers. Now, if we have a large area, 20 square kilometer is a quite large area. So it is not that easy. We're going to count each and every species and their numbers, right? So if this is a task we have to do, we have a method for this ecological survey. So method come on the quadrant method. Okay, let's try to understand how we do the quadrant method. So when it comes to a ecosystem, let's assume one ecosystem is like this. And the second type is like this. Okay, this represents generally the two type of ecosystem. So what is different of these two type? So if we consider this area, if it is like a grassland, so this grassland is mostly filled with the grass and a little bit of a small flowers. This one is a mountain area and starting with the small river, and from this area there is a slope, and we can observe many different species, and their distribution is also very different. So their distribution is also very different. In other words, this is well uniform area, and this area is very irregular area. So A is uniform, B is irregular. So if we wanted to do ecological survey in these two different areas, uh, we have two methods. For this one, we use random sampling. For this one, we use systematic sampling. random sampling and systematic sampling. Okay, now if we do a random sampling and systematic sampling, we're going to do like this. So let's assume the area is like this. And this area is going actually the upward like this, but still the total area is like this. Okay, then the both areas we're going to do some method. So we're going to make a small map of this area, and we're going to add certain information to the map. First, we draw the map by measuring this area, and after that, we can add grid lines to the map. And also we can add grid line in this direction. So let's say this is a grid format. And from the grid format, we can give values to each grid. Example, if I take a system like labeling them from 1, 2, 3, 4, 5, 6, 7, 8, 9, and the other side I can give A, B, C, D, E, F, G, H, I. So each coordination has a value now. For example, this one is a B2. This one is a E5. This one is a H6. So uh, we can give a value for each name for each grid areas. So map the area, place the grids, provide or give names for grid. When we have all the names of the grid, we're going to use a random areas. We're going to pick up random areas, right? Pick random areas. So how do we do that? We can use a random number generator. We can give random number generator. So all the number feed to the programs, and then we can provide us this like 20 random numbers. So after we get 20 random numbers, it would be like this. So it gives the system gives this, 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 this. So I'm giving these other random numbers now. So when we have random numbers, we're going to count the species only in random numbers areas. So species we are counting only in these areas. Then count the species only in random numbers. Okay, we will see for both method what is the procedure of counting. Now, systemic sampling. Systemic sampling, what we trying to do, we trying to place lines like this. So this is the line. So in this line, I'm going to mark a fixed distance. For example, here to here, it's 1 kilometer, and 1 kilometer, 1 kilometer, 1 kilometer, 1 kilometer, like that. So this is a fixed distance. And again, I'm do here like this. So place lines, mark a fixed distance along the line. And I can do another different way. So instead of this, I make a belt, but there's not much different actually. This is a belt. Either way preferred, I can do it. This is a belt. And from the belt, I'm also going to mark a fixed distance. For example, starting from here and here and here, 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 here. So this is a one area, this is a one area, this is a one area, this is a one area. Right. If we do any preferred method, this is called line transect. This method is called belt transect. Line transect and belt transect. So I can mark in my map the line transect and the belt transect. Then once I complete for this kind of ecosystem for random sampling and this kind of ecosystem for systemic sampling, I use only one method for counting. What is the method of counting? I use a quadrant. Then for both, use quadrant for counting. For both mean this and this. Now, what is a quadrant? Quadrant is a square shape wooden, plastic, or metal, whatever, uh, just like a frame, right? This is just like a frame. And then inside, there is also some divisions like this. It could be 1 meter, 1 meter. So then I take this to the field, right? And for each case, random sampling or belt or line transect. So if my random number is like this, so random sample number example like this, and this, and this. And for this case, line transect or belt transect, it is like this. Then I keep this quadrant in this place and count the species. And for this one, again, keep and count the species. So then quadrant can be used to count always fixed area in each place. So after counting, get the mean value. Find mean value to find total. Mean value multiplied by total number of quadrant. Quadrant mean like total number of these coordination points, right? Let's say in this case, we take sample 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So we get a mean value of 13 sample. And how many we have? Roughly 9. 1, 2, 3, 4, 5, 6, 7, 8, 9, like 81. So it's 81. So mean value multiplied by 81 to get the total number of species. Then there are a few other things. If species density is high, so for example, let's say small plant, so it's very difficult to count 1 meter range because density is very high. I mean, if you look at the grass, so 1 meter count in the grass is again difficult because it's too much. So then we can use a scale. So A, abundant, C, common, F, frequent, O, occasionally, and R, rare. It's called AC4 scale to count the number of plant. Like if there are so many plants in one place, we give the mark A, and there's the less, we give R. So then we can still give values after that. Then this we add to the numbers to calculate. For example, abundance refers to like 100, and rare refers to a 5. So within that range, we make a scale. So we just count how many grass in 1 meter, and then abundant it has 100. Then we can make some kind of 75, 50, 20, 5 like this. And it is same for the other one, random sampling and systemic sampling. Both method is same at the end. Only based on the ecosystem, if it is irregular, we go systemic sampling. It is regular, then we go random sampling. So after done this one, we can try to find same method that the quadrant method used to measure the species. Okay. Now, another thing we are going to learn here in the 11th practical. So we have done two practical. Let's say we have completed two practical. Usually we can complete uh, two practicals in a class. So that basically then 5 to 4.
So can you like send that file, that
Yeah, I
PDF?
save this one. I said what I will WhatsApp this one. So
So
there's
can you
another
show the about
uh,
one?
this one
Yeah, what's the topic of the investigation?
here, quadrats and transect. So quadrant and transect. So quadrats and transect to determine the distribution and abundance of organisms. These are the practicals. Quadrant and transect to determine the abundance and distribution of organisms. It is come under the biodiversity. To find the species richness, we have to count the organism. So we use this method for ecological surveys. And here is the principles. If we have a uniform area, then we use random sampling. If we have an irregular area, we use systemic sampling. Systemic sampling, again, we divide into line transect and belt transect. So transect method we use for the systemic sampling, and the random sampling we use this still it's called quadrant method, but at the end the quadrant is the one we use to count. So both cases, we take the quadrant and place in the given area and we count the species. Okay. Now we have something important here to learn. When we count organism and try to find the relationship, we need a method to analyze the data. For that, we have to use the statistics. Now, until AS level, we mostly based on a general statistics method to find whether it is significant or not, right? So this word significant is very important, and if you remember what we actually did, so if we have a graph like this, and this is the second one, to compare A and B the values, we use the error bars or standard deviation for the comparison. For example, if error bars are not overlapping, then we concluded it is not significant. So error bars or standard deviation is not overlapping. Then we concluded significant. Another instance, uh, if we see error bars are overlapping, for example, this, overlapping, or standard deviation overlapping, then our conclusion is not significant. So this is the method we use AS level to find a significant or not. Now, again, what is this significant? Why we define this word, and what is the value of this significant? Why it is so important statistics? We have again some kind of understanding. That means if we look the probability, we can make a few conclusions. Anything can happen. There's a chance. So there's a chance anything can happen due to a chance. So when we make this kind of conclusions, if someone is doing an experiment, we can also apply the same principle. Yes, you got some result. Your result are chance. It's a probability. So you have done your experiments 100 times, and after 100 times you got some result. This is due to a chance. This happened due to a chance. So that is the problem we have now. I mean, let's say simply like this. You take 100 people, and one group, you are just giving a water and saying this is a medicine and this can cure your disease. And the other group, you are also giving again water with different color. Just colored water you are giving as a medicine. Now you can understand this. This has some effect because when we learn in the clinical trials, the placebo is a very important group because we learn we have to anyway do in phase 3 trial the placebo study group, because there is some psychological effect. There is a effect of the drug we are giving. So this is one example how certain things can still give some effect because of the chance. So we have only one solution to overcome the problem. If
So in that other one, error bars, standard deviation is not.
error bars, I mean, and standard deviations we'll consider as the one thing, because uh, the standard deviation is the right word. I think standard deviation is right word, and sometimes in AS level we use error bars. So error bars and standard deviation still we have to use, because the exam sometimes we have seen the words are using here and there. But at the end, it is the same thing, only how we show and make it is the different. Like how we calculate is different. Still show the variations. Both shows the variation. So we make better understanding, both are same. The difference is how we calculate it. So error bars or standard deviation are same. So here, what you are seeing in this graph, a standard deviation or error bars. Think at the moment they are more or less same. It's exactly same. Only different is how we calculate. Based on the calculation, the layer length may be a slightly change. Otherwise, they are same thing. So error bars is not overlapping. Then we call significant. Error bars are overlapping. Then it's not significant. So this is the method we use mostly AS level. But when it comes to the statistics of the higher level of statistics, we use another method to analyze it. To analyze it, the significance is it's a chance. So we are trying to understand the probability, any event, there is a probability. So anything can happen due to chance. So what we do, we bring this chance to 5%. So we bring this chance to 5%. That means, yes, this can be happen due to chance, but we minimize the chance to 5% or 0.05. That means we take 95% or 95 is due to a reason. So this is chance, this is a reason. So this is something like a good indicator. So we have to make a method now to bring the probability to 5% and others to 95%. So scientists develop a statistical test based on this concept. That means we are not much rely on this overlapping, not overlapping kind of theories. We're going to do a statistical test. Based on the statistical test, we make a conclusion, whether it is significant or not. So still, we going to make a conclusion, it is significant or not, based on the statistical test. This is the much standard method. So this is something you have to understand. You are moving from A's to A2 and still you carries the same concept but different way. So error bars overlapping is the method you used, AS level. Now you're going to perform a statistical test to make a conclusion of significant or not significant. When it comes to significant or not significant, how you can explain significant or not significant based on this. So anything can happen due to a chance. If we do an experiment, we get a positive result due to a chance. But we make a statistical test to minimize the chance to 5%. So we can't bring it to 0, we can bring it to 5%. So when we see the chances are less than 5, then we accept, yes, this has some kind of a scientific basis. This reduction is due to a scientific basis, not due to random or due to a chance. Let's say this is the drug used for the uh, hypertension, and this is a control, and this is a drug. And when we use this drug for hypertension, reduce the blood pressure, we can see reduction. This is the reduction. We don't see 0. Like if it is control is 100, drug is 0, I think then we don't want to use any statistical test. So it's a black and white. Control is 100 and this one is 0. But we have values in between. So it is not 0 and 100. We have values in between. For example, the control group, they give a placebo, and the blood pressure is, let's say, 130 mean value, systolic blood pressure 130. And for this one, after giving a drug, it is 120. Systolic blood pressure is 120. So then we have a question: is this a truly effect of drug, or is this a chance? Because many factors are involved. People are different, and they are still genetically different. So many factors are there. No? So if we do this, if someone is doing with 100 people, luckily this company got people with some kind of a genetically different people. So these group, they have some kind of a effect, like a placebo effect, like things, it work. But still we want to do a statistical test, whether this is truly scientific or chance. So that is the basic for the statistical test. So we have to learn basic concept of statistics and basic concept of three statistical test. We're going to learn t-test, chi-square test, and correlation test. T-test, chi-square test, and correlation test. That will take some time. Learning practical is not time consuming, but just to cover up the statistical part, it takes time, because second question is mostly based on the statistical test. For example, the same paper, we have done first question. The second question, no, it's third question, right? The second question is, this paper has, I think, 4 questions. Third question is based on the statistic. Here, for example, calculate the correlation coefficient using the formula. So we have to use this correlation test to understand and write answers. Third question. And fourth questions is a detailed experimental method. So that means this question is 8 marks. You have to devise a method for this experiment with detailed description. You have to write a complete report about the experiment. I mean, 2 pages are given for that, and some kind of analysis. So first and second in this paper is similar to AS. Third question is the statistics. So to learn, to do this statistic question, we have to complete the statistic parts. That is the format of the paper. The format doesn't change much. Sometimes 3 questions, sometimes 4 questions. If you get a 3 question, second question is statistics, third question is detailed report about the experiment, first question is AS type. If you get a 4 questions, first and second looks AS type, third statistics, fourth detailed descriptions. So that is the format of the Unit 6 paper. Right.